In this video, we will discuss about the matrix stiffness regulates the pathways that helps in tumor proliferation and metastasis. So, how this stiffness affects the cellular pathways? We will discuss the brief summary that is presented in this review paper. So, what it says that cancer cells are, as soon as the surrounding or stiffness around the cancer cells increases, it leads to upregulation of some pathways and downregulation of some pathways. In upregulations, it will lead to the upregulation of ROCK pathways, CXCR4 pathways, MAP kinase pathways, GMD-DA1 pathways, MRLC pathways, and some of the pathways we have discussed in this channel. You can refer to those videos. For example, this increase in the ROCK pathways will affect how the ectomycins work. That it will make the ectomycin more stretched out, and that helps in the proliferation of the cells. And similarly, the CXCR4 pathways also helps in this EMT, this epithelial to mesenchymal transitions. And there are some pathways. So, however, apart from those that are upregulated, there are some pathways that are downregulated also. So, yes, UBTD1, RAP2, and overall effect of this will lead to this contraction of the cells. For example, more precisely, this upregulation of ROCK pathways will lead to the upregulation of MRLC pathways, and that will lead to the contraction of the cells. And these contractions, along with the other changes in the pathways, will lead to this YAP-TAZ upregulations. We have discussed the effect of this YAP-TAZ upregulations in our previous videos, where the upregulation of this YAP-TAZ will lead to the more nuclear localizations of this YAP-TAZ, and it will lead to the epigenetic changes in a way that it will promote proliferations, migrations, or metastasis, or invasions, and it will also help in this drug resistance and the stemness of the cells. So, what overall the general picture come out from this summary is that the cancer cells prefers stiff matrices, as it will not only act as a physical barrier to prevent them against the immune cells or the drugs, but it will also lead to the epigenetic changes within the cells in a way that will make the cells more stemless in terms of the behavior, or make the cells more mesenchymal type, or that will help, or not harm, that will help the cancer cells to metastasize across different parts of the body. And that is why, in general, the stiffness around the cancer cells are usually lower compared to the case where cancer cells are usually found, or most precisely, the solid tumor cells are usually found with very stiff matrices. And that is one of the reason from the experimental point of view, that when someone sees the cells, they usually use the ROCK inhibitors, so that the cells ectomycin can relaxes and cells can stick well to the stiff matrices. And after and these ROCK inhibitors are usually used just at the time of CD. After that, it is removed, so that ROCK inhibitors will start to work accordingly. Thereafter, other things that we need to understand about this figure is about the YAP-TAZ. The YAP-TAZ in normal form are present in the cytosols. However, during the cellular changes, this YAP-TAZ becomes unstable, and they will translocate into the nucleus, where it will trigger these changes that will favor the tumor growth and proliferations. So this is a brief summary how the matrix stiffness helps the cancer cells, not only in terms of the immune evasion, but also make the cellular changes in a way that will make them more aggressive and proliferative in nature. So that's all for this video. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.